Muhammad Umar Farooq biological video. Please make sure to subscribe, like and share. Ask the question for answer and comment about the artwork. You can also answer question to each other for solving the problem. So here today we will discuss about the thymus gland. So the thymus gland is the gland because it is the endocrine gland because it's produce a chemotactic agent. So that is a stimulate to another tissue or organ to tissue to recruit the cell. So that is why it is the gland and it is endocrine gland, and it is the used for the T cell maturation. So here we will discuss just the T cell maturation in the thymus gland. So the thymus gland is the part of lymphatic system in which T cell educated for attack against infection site. So the thymus gland is a primary lymphoid organ, which used for the maturation of adaptive immune cells. So the adaptive immune cell is first here is a person which contain a heart and lymphatic system, and that is the other gland is also other tissue is present, which that is the lymphoid tissue. So the spleen and the tonsil is the secondary lymphoid tissue or organ, while the thymus gland is the primary for producing a immune cell activation. So here is the tonsil, and it is also part of that. But here we will all just understand about the lymphatic system, about the basically the thymus gland. So here is the structure of thymus glands. It look like this, and it is the two lobe. It is the behind this front lobe is the right lobe, while the left lobe is looking like here. So I am drawing the diagram is like that. So here is the left lobe and right lobe, while the vein and artery are present and lymph duct is present in the figures you can see. While here the cross section of the thymus gland is here, which that is made up of medulla and inner cortex as well as outer cortex. So remember, so let's talk about here is the bone which contain a stem cell. Stem cell mean it is produce the pluripotent stem cell as well as it is divided to again produce stem cell by the mitosis, while it is also produce the functional cells, for example the hematopoietic precursor, which that produce the white blood cell and red blood cell and platelets. So that is why it is stem cell which convert into the pluripotent stem cell, and that's hematopoietic precursor will go into the blood vessel. So here is I am drawing the blood vessel. So the lymphoid progenitor will be formed is here into the blood, and it's go into the thymus gland. And so you should remember that thymus gland is made up of three partitions. So inside the medulla, outer is the inner and outer cortex. So here is the structure of thymus gland I am drawing, which that we will able to understand the maturation of the T cell and activation of the T cell. So here is the outer cortex, while inside the inner cortex and upper part is the medulla. So inside is the cortical medullary junction is present in the red color, while inside is the medulla. So let's talk about it from here. That is the cell lymphoid progenitor will be enter into the thymus gland, and that's important thing is here is the thymocyte or cortical epithelial cell is present. This cortical epithelial cell produce a chemotactic agent, so that is looking like a hormone or ligand. So it is the made up of thymosin, thymotaxin and thymopoietin or thymic factor. It's collectively so basically called is a thymic factor. So this thymic factor will activate to the lymphoid progenitor from the bone into resorption into the basically blood vessel and go into the lymphoid progenitor into the outer cortex first. So when we'll go into the outer cortex, that structure is look like this. It's contain DNA, so that means that contain nucleus, and is the RAG1 and RAG2 is the factor which that stimulate the DNA to transcription factor by the binding of transcription. This is the binding of transcription factor, RAG1 and RAG2, which that bind, and it's here is the here is the double negative receptor containing cell which that activate of gene transcription to translation, so to produce the TCR, T cell receptor. So this cortical epithelial cell will stimulate it, while here is the T cell receptor will be produced by this protein translation, and when go into the double positive cell will appear by the thymosin factor will stimulate more to produce TCR is according to the epitope R antigen, while it is also produce the CD receptor or CD cluster differentiation protein will be arise. So this CD is in the form of precursor of the CD4 and CD8. So we will not understand about the CD40 etc. So just we should understand with the basic concept. So here is the T cell receptor is fine, and that is the modification of this T cell receptor is according to the epitope or antigen. So this antigen, for example, is according to the TCR will be modified by the DNA transcription to translation. So here is the CD cluster differentiation protein. So this cluster differentiation protein will be differentiate into the CD4 and CD8 molecule. So this marker is the CD4 and CD8 marker. Right? I hope you understand about this concept. We will discuss about the maturation and their development. So it is going to development. So remember about that. It is the during child to adult, it is more efficient work of the thymus gland, while the older age will be diminish the function of this thymus gland. So here is the selection positive will be occur if the epithelial cell will be receive the MSC class 2 and MSC class 1. MSC class 2 molecule will be bind with the CD4, while the MSC class 1 will be bind with the CD8 to mature it, and that T cell receptor are present, and this T cell will be either go into the negative selection or positive selection. So let's talk about the negative selection. Negative selection mean when the self peptide not recognized because damage our own tissue by recognition. So it means the cell will be, first of all we will discuss about the positive selection. So here is the MSC class 2 and MSC class 1 molecule which that's bind with the CD4 and CD8 respectively, and T cell receptor will not bind with the self peptide of the medullary epithelial cell when go into the medulla. So it means the self peptide not recognized because damage our own tissue by recognition and produce autoimmune disorder due to the recognition of itself of our own self of the cells. So it means it's produce autoimmune disorder like Graves disease. So it means here is the autoimmune disorder if TCR recognize any cell containing peptides. So it will be not, and it means the positive selection, while the negative selection mean the TCR will not. TCR will be negative selection will be TCR will be bind with the MSC1 and MSC2 by the recognition of CD8 and CD4. So that means the thymic cell will be apoptosis due to the recognition of the our own cell. It means that our own cell recognition by the self peptide can be damage our own tissue. So that's why it is apoptotic cycle. So it is negative selection. So let's talk about here is the medullary epithelial cell, which that bind with the CD4 just only with the MSC molecule 2, and that means the CD8 will be not bind, while on the other hand CD4 will be bind with the MSC class 2, while the CD8 will be bind with the MSC class 1 to up regulation of CD4 molecules and CD8 down regulation. It means on the other hand up regulation of CD8 molecules and CD4 down regulation. It means the single CD cluster differentiated protein will be formed in the single cell to form a T helper cell for MSC2 recognition, while on the other hand cytotoxic T cell for MSC class 1 for the another recognition. So the cytotoxic T cell is directly kill the damaged tissue damaged cell, while the T helper cell is the recognize the antigen to give the signal for recruit the macrophage as well as monocyte, etc. So it means here is the recruitment of that. And on the other hand, this cell is bind with the MSC class 1 or 2 with a dendritic cell and with a dendritic cell to recognize that antigen. It is antigen representing cell, which that antigen present to the macrophage, and there's macrophage to the another cell, while the toll-like receptor are present in the macrophage to recognize the pathogen, right? Here we will discuss about the adaptive immune system. What is the relation with the T cell maturation and their adaptive immune system? So the adaptive immune cells are T cell and B cell. These are the part of second line of defense in immune system. So the adaptive immune system is in two types: humoral response and intracellular response or cell mediated response. After the maturation, this T cell will go into the lymphatic system, spleen, or tonsil, and lymph node for going into the inflammatory site. So here is the adaptive immune cells are T cell and B cell. These are the part of second line of defense in immune system. So the adaptive immune system is two type: humoral response and intracellular response, or cell mediated basically response and humoral response. So let's talk about here is the humoral response. So the humoral response means the produce of antibodies. So the production of antibodies by the recognition of the antigen through the antigen representing cell or macrophage to the T helper cell to produce chemokines, and this chemokine will be attract to the naive B cell to produce that antibody, while it is also recognize B cell with itself of the antigen to produce the plasma cell and T memory cell. This plasma cell and T memory cell produce antibody. Plasma cell is quick response, while produce immunoglobulin or antibody, while the T memory cell will produce the in the long term, which that again infection to activate. So it means the production of immunoglobulin by this mechanism. So cell mediated response mean that is the cell mediated response is the T cell is cytotoxic T cell, which that is naive T cell will differentiate into the cytotoxic T cell after the recognition of the antigen. This is the tissue and damaged tissue by the viral infection inside of the cell to represent like a antigen representing cell to represent the piece of the antigen with the MSC class 1 molecule. So it means the TCR T cell receptor will bind with it, while the CD8 molecule will attach with the MSC class 1 to activate and produce perforin and granzyme, which that invade inside move inside to the inside of the cell and will become a cell death. So that thanks.